What is yours?
Hey, thank you very much, Jean-Pascal, for the invitation to speak in what I'm sure will be a very interesting session here. At least the last three talks will be very interesting. Okay, before I get going, if I can get going. Okay. I want to make sure to acknowledge those who did the work I'm going to be talking about today. The modeling work that is the bulk of the material I'll dwell on is a project that's been going on really for quite a while now, because it's been gone through a few different stages of unfolding over the last few years. And it's a project being carried out by Leah Dunker, who's a PhD student here at Gatsby with me, and Dan O'Shea, who was a graduate student, now a postdoc at Stanford with Krishna Shenoy. The data we're going to look at were collected by Dan and by Werapong Gu, and they were working in Krishna's lab. And this is part of a long ongoing series of collaborations between Krishna's laboratory and mine, which have been very rewarding. Okay, I'm going to talk about that data set, and we'll sort of dwell on some details there, but I'd like perhaps in the back of your minds for you to be carrying with you a sort of a larger question as we look through these particular data. And that is about sort of the nature of models. Models are always an approximation to the truth, but we can sort of build these models when we're actually trying to describe neural phenomena in at least two different ways. And I'm going to choose two here, but obviously there are other things one might think about. One approach, and the approach actually that my group has spent a lot of time working on, are things that you might call phenomenological models of functional data. So we take data recorded in intact brains, often awake and behaving brains, that data might look like a series of spike trains, such as this, and try to infer from them some sort of portrait or description of what's going on inside the circuit, or perhaps a dynamical flow field, which describes how the system will evolve over time. And this particular picture I took from some work that, again, Leah was the first author on, that was published at ICML last year. So these sorts of models are typically statistical in their form. Right? We write down a set of equations that are relatively abstract, but then they have a lot of free parameters in them. We're trying to adjust those free parameters to describe the data. There are probabilities involved, partly because at the level of description we're working, we regard the system as noisy, partly because, as I said, models are approximations to the truth, and having probabilities allows us to titrate the sort of relative accuracies of different pieces of the model. And I think these models can be quite powerful. So when we look at populations as a whole in this way, what we're looking for is coordinated activity in the population. And I think frequently that coordinated activity captures something that seems to suggest the nature of the in vivo computation. So one place where we've done a lot of this is in the motor cortex, where those sorts of computations seem to be the unfolding of some sort of dynamical process. Sometimes in some of Mark Churchland's work has been described as rotatory dynamics, that basically creates the dynamical substrate for the control signal that moves the arm. Or at least that's the hypothesis. The other approach to building models at this sort of broadly at this level is the mechanistic one. That is, we start with a description of the circuitry and try to make sense of or try to understand how those components will act together. And this can be, a very biophysically detailed sort of model, the sort of thing you might set up in a Genesis or neuron simulator or in the blue brain. Or it could be abstract. It could be, a mean field treatment of a bunch of point neurons. But in either case, we're sort of taking some sort of abstraction of the experimental findings about the components and trying to understand their properties. And as a result, what we get are effectively constraints on the activity that come from the substrate. Okay, but it's not impossible, but it's relatively hard to see how these directly create the sort of subtle, complicated computations that we might be seeing when we measure data in the real brain and are trying to capture in these statistical and non-mechanistic models. So the obvious question, and the one that I want you to have in the back of your mind, is how do we put this together, right? How do we take these mechanistic constraints and bring them to inform the phenomenological models of computation? Or if you like, get the other way around, how do we use some phenomenological data and statistical methods to inform sort of details of the parameters in the mechanistic models and try to understand, and therefore try to build something that gives us both a sort of circuit level bottom-up sense of what's going on, that is how the components are arranged to create the results that we see, but also a sense of the computation that allows us to match the details of the ongoing neuronal activity. And I'm going to suggest one way in which one very simple constraint, Dale's law, can be used to do some of this. So we'll sort of discuss at least one I'm not quite sure what to call it, but a direction in which this we might be able to explore further. Okay. Let me introduce the data that we're going to look at. These are data recorded in the motor cortices of non-human primates who are doing a delayed center-out reaching task. So in this sort of task, the animal's asked to initiate it by touching a point of light on the screen. A little while later, a second point of light is illuminated somewhere on the screen, in this case, one of 4 locations, as we'll see. And the animal has been trained that that's a point that it will need to touch, but not just now. It has to wait a little while. After a while, that's the delay part of the delay center out. After that delay period, there's a go cue. In this case, that's the extinction of the center target. And at that point, the animal knows, okay, now it's time to reach over, touch the other target. And if it does so rapidly enough and accurately enough, it will get a reward. Okay, in this experiment, Dan and Weerapong had used a viral construct to express in pyramidal cells in the motor cortex a excitatory opsin. So this is a protein in the membrane. It's a channel. When you shine light on it, it will excite the cell. The specific opsin was C1V1. And during this while the animal was performing these center-out reaches, they would turn on a laser and shine light on a group of cells, either at various points in time during the trial. And there are 2 of them illustrated here, but we'll actually look at data from 3 of different epochs. So one of them is during the delay period, while the animal knows where to reach but hasn't actually reached yet, or is withholding the reach. One around the time of the go cue, and it takes a little bit of time for the physical go cue on the screen to be processed by the system and actually start to, the cogs in the motor cortex to start to turn. And so this potentially might interfere with initiation of movement. And then there was another epoch which was during the movement onset. We could see what the effect would be during the movement onset. During the movement itself, rather. The transfection and the light delivery and everything were all perfectly successful. If we look at, they recorded simultaneously both at the fiber that was delivering the light, that's the optrode here, but also up to 3 millimeters or 4 millimeters in some cases away. And you can see this is the change in firing rate as a result of the stimulation. And you can see very substantial changes in firing rate. I draw your attention here, this is to the scale, it's 150 odd spikes per second for a large number of these cells. Sort of median numbers would be somewhere around 50. So large changes in firing rate, many of them polysynaptic because they're some distance away from the fiber, and some of them are suppressive, which, given that this is an excitatory opsin, will be polysynaptic. And over a very substantial piece of the cortex. So this 3 millimeter diameter radius region, this really takes up a fairly large part of the cortical surface that seems to be devoted to controlling the arm. So this is sort of based on the standard homunculus. So it's a large effect. Nonetheless, the effects on actual behavior were very small. Unlike an electrical stimulus, turning on the light did not evoke a movement directly. If it was shone during an ongoing reach, there was no change in the trajectory that the reach followed at all. The one effect they noted was that if the laser was turned on around the time of the go cue, the monkey was very slightly slower to initiate the reach by about 10 milliseconds, but that's for a 200 millisecond sustained activation around the time of the go cue. So it's still a very small and subtle behavioral effect. Okay, so what's going on, right? There's a big change in the firing in an area that we think is intricately involved in the initiation and execution of these movements. Why did nothing happen? Well, one possibility is that this sort of hammer effect of just sort of pushing on all of the excitatory cells in a local area is just sort of irrelevant to the subtle patterns of activity that dictate the task. And so one way to look for that is to use these sorts of statistical techniques, in this case a really baseline, very simple one, PCA, principal components analysis. Just look for the sort of the bulk signals of coordinated activity in the area and ask, well, does the stimulus affect those or does it not? And so here are the top 3 principal components, the time series of those components, as the animal makes reaches to 4 different targets. That's the 4 different colors that you see here. And this is the activity during the delay period here. This is the time of the go cue. You can see that there's some encoding of the target location during the delay period. That's well known. At the time of the go cue, there's with some delay, there's an initiation of a change in activity here, and then a little while later, you see the movement of the arm actually begin. That's the movement onset here, MO, and then this dynamical pattern kind of carries through throughout the movement. Okay, so the question is, does the stimulus affect this pattern in some direct sense? So let's look to begin with the first principal component. I'm going to, as I said, look at 3 different epochs of stimulation in the delay around the time of the go cue and the time of the move, the movement itself. That's the green shaded area in each case. As you might expect, on a trial where stimulation was about to be delivered, but it hasn't been delivered yet, that looks exactly the same as a trial in which there was no stimulation. So that's reassuring. Turn the laser on, and there is a big perturbation in this leading principal component of the activity. And remember, the principal component's been defined by unstimulated activity alone. It did not we didn't use the stimulated trials to define those principal components. The laser stays on for 200 milliseconds and activity continues to evolve. And it's worth noting, and this will be relevant, that some of that evolution resembles the evolution, but with an offset, that we would have seen had this been a normal unstimulated trial. So the dynamics are still going on underneath this stimulus, and that is an important point. Turn off the laser, and very rapidly, the activity returns back to where it would have been had there not been a stimulation. And basically with almost no perturbation, it's a very, very subtle, short perturbation, which I won't have time to go into, but is interesting and can be modeled. Things just return to where they were and then proceed along the same dynamical trajectory as would have been followed had there been no optogenetic perturbation at all. Okay, same is true for the other principal components. So stimulation does perturb the task-related activity, but the activity recovers very rapidly and with very little effect on the future evolution. So in somehow the dynamical system is robust to this big kick in firing rate space. How? What's going on? So of course, one possibility is that the neurons that Dan and Weerapong recorded weren't actually part of the dynamical state of the system that drives that's part of the dynamical system that creates this activity. That's possible, but I think there's lots of other reasons to believe that at least some part of the dynamics that we're looking at in the motor cortex do originate there, or that the motor cortex is a component of the dynamical system. It may involve thalamic loops, but that the motor cortex isn't just reading dynamics that came from somewhere else entirely. So the other possibility is that somehow the dynamical system ends up being robust to this kind of perturbation. And that's what we're going to investigate, is whether that makes sense. And so just to unpack what that means, if I were to write down a dynamical system for the sake of argument, a linear dynamical system that tells us how the firing rates of the whole bunch of cells, that's a vector nu t, evolves over time, and I'm going to use a discrete time formulation for this entire talk, so we'll always always write things as vectors at different points in time. So we have this linear update, because I said I'm going to assume it's a linear dynamical system. There's some input signal, which, for example, would initiate the movement. If we add to that the optogenetic perturbation, the stimulus S, then effectively what we need is, as that stimulus runs through the dynamics, it decays away to zero very rapidly. It doesn't create its own ongoing complicated dynamics. Okay, and so in effect, this is a kind of null space. I mean, we've written it as explicitly a null space of W. It doesn't have to be quite that, but somehow the dynamical system created by W needs to cause S to decay away rapidly. And the question is, well, I mean, that's one way of describing the result, but why might that be true? And this is where the mechanics come in, and in particular Dale's law. So let's think about what an EI network that needs to generate robust computation in the presence of noise might look like, or what constraints would that impose? So if we have noise, a simple assumption for the noise is just sort of random input fluctuations with equal variance in all directions, so unit variance normal. In this case, if we pick an arbitrary unit vector u and ask what's the variance of the noise that we will see after one step of dynamics in direction u, the answer is given by this quadratic form with WW transpose in the middle. And this means that the direction that will carry the most variance after one time step is the one that is the leading eigenvector of this matrix, WW transpose. Now, WW transpose is the connectivity of a network that we're going to assume satisfies Dale's law. So it has a pattern that looks like this. If we order the cells with all the excitatory ones followed by all the inhibitory ones, then if we look at the columns, they're all of one sign. And if we look across a row, we have positive entries and then negative entries. So the colors here correspond to the signs. And if you take WW transpose for a matrix like this, if you just think for a moment about where the signs add up, or where they multiply rather, you end up with a product which is all positive. Okay. And there's a useful result, the Perron-Frobenius theorem, that tells us that the leading eigenvector of such a matrix has all entries of the same sign. And the sign, of course, is arbitrary, but we'll take it to be positive. Okay, so that's one observation. And so what does that mean for noise robustness? Well, it means that if the noise after one time step is going to be mapped with greatest variance onto a direction that is, not exactly a common mode, but broadly all the cells moving in the same direction, then it's necessary for that mode to, for the noise along that mode, or for that mode, and therefore the noise along it, to be suppressed. And so one way to do that, of course, is to have, if this is roughly coactivation across all cells, if W then is balanced such that the net excitation roughly equals the net inhibition across each one of these rows, then this will decay away. And so this sort of gives us another reason, if we needed one, for why it might be useful for systems to be broadly balanced. Okay, so Dale's law and robustness sort of pushes us towards this idea of balance to eliminate variability that gets squashed into this kind of net common mode. What about the stimulus that we gave here, a positive input that targeted not all cells, but excitatory cells specifically? Well, if we took an input like this, that is in the top half of the vector, so it's hitting the excitatory connected cells on this side, and ask what the effect of that will be, if roughly the excitation that drives all of the cells, that is the sum of just the excitatory part of each of these, is sort of roughly similar, that will map onto one of these modes that's sort of general common activity. And so in two steps, we would end up rejecting this kind of noise. It's not an immediate rejection, but it's sort of a two-step rejection. Okay. So that's sort of a little bit of very hand-wavy argument for why you might expect an EI-like network to be somewhat robust to this kind of perturbation. But that puts aside this observation that we have a network that can generate very slow systematic activity over time. I'm running out of time, so I'm going to very quickly try to say what we did at a very high level. So we built a model in which, and when I say we, I mean they, Leah and Dan built a model in which we have a vector of firing rates, which we think of as a population of underlying cells. These will be excitatory and inhibitory, as we'll see in a moment. And we wrote down a linear dynamical system with an input. The input just initiated the movement. So it was a very simple input, and some noise. We sought to explain the measured data, which I've called Y, through the projection of nu through a matrix, actually a random matrix J. So C is a free parameter that we get to fit to explain the data. J is just a random projection of nu. Okay, and this captures the fact that the coordinated activity in here seems to be low dimensional when we try to fit it. The connectivity, as I said, satisfies Dale's law. It had a form of EI balance. We didn't actually impose the balance. We just had noise in the system, and for the arguments that I just made, that tended to drive it towards being balanced. And we also said that the dynamical space that implements this should in some sense be decoupled from the rest of the network. That turns out to be this particular constraint. I won't step us through it right now. But basically it says that stuff that's orthogonal to the subspace defined by J shouldn't interfere with the task. Okay. So this is a model that we can now fit to the data. We can adjust the parameters W and C and some aspects of U and D in this model in order to describe the data. And we fit the data to the PSTHs recorded without stimulation. Okay, so no optogenetic perturbation here. These are the top three principal components you saw before. This is the model fit. So it's perfectly capable of capturing that dynamical process so what we have ended up with is an EI circuit that creates these these slow profiles. I will skip, because of time, a discussion of the eigenvectors of these and go on to show what happens when we actually look at this network, which has not seen the stimulation input in training. We've adjusted the parameters without that. What happens when we turn on a stimulation vector? So we add an S, which is a random perturbation. So basically the ideal delivery targeted all excitatory cells, but of course the Upson expression will be noisy, and the infection will be noisy, and then the light is delivered through a fiber, so it will only affect a localized set of these things. So that's the stimulation vector we'll use. What happens? And so, as you can see, the model is perfectly capable of capturing this offset due to the stimulation. Okay, there's we have to learn the mapping from the stimulation vector to the neuronal activity, because that was not fit before, but we don't relearn the dynamics at all. So the dynamics of the system were learned entirely based on the unperturbed stimulation. Nonetheless, dynamically, as soon as the stimulation is removed, we do see exactly what we saw in the data: return to baseline, and then things just carry on as they would have otherwise. And that's true for delay early here. It's true for stimulation during the go phase, when you can see, as I pointed out before, that the underlying dynamics do continue through the stimulation, and it's also true during the movement phase. Okay, so the model responses match recorded data, and it looks as though one possible account for the robustness that we see in these data is because of the EI structure, and that if that has to be tailored to reject noise, which would otherwise be amplified into this common mode, then it also ends up leading to robustness to stimulation. Okay, let me just end. So what we've tried to do is work out a way to build a statistical model for data that embodies some or incorporates some of the circuit constraints that we know exist, and a very simple one here, Dale's law. We did so using input from neural data, and in particular, in this case, neural data which had a perturbation that came in some sense in circuit or component coordinates. And so that forced us to think hard about componentry. And as a result, we thought a little bit about sort of the structure that we might expect in these sorts of networks when they need to create slow systematic dynamics and therefore and have that and be robust to noise in that dynamics. And so these two pieces of information informed the way in which we were able to build this network, and show that in the end, I guess, build a model, an approximation to the truth, that is a circuit with certain anatomical constraints. It's a system of excitatory and inhibitory cells that is able to create the sorts of dynamics that we see, and almost for free ends up rejecting the stimulus in exactly the way that the real data did. All right, let me remind you again of the people who did the work, thank the funding sources, and I can take some questions.
Okay, great, excellent. Thanks, Maneesh, for this excellent and really stimulating talk. Okay, so for the moment, there's still no questions in the QA. So I just remember all the attendees that they can put some questions in the Q&A form. And meanwhile, maybe I'll just ask some questions, if I may. Mm-hmm. So if I understand correctly, you're fitting a linear dynamical system, right? With the hidden variable, and you have your matrix W. Mm-hmm. It's all nice. What about nonlinearities? Would you expect fitting a nonlinear model to provide any additional insights? Or
so it's an interesting question.
Comment on the.
Yeah, no, yes. I think obviously the real system is nonlinear, and so the linearity is one of the ways in which the model is an approximation to the truth. I think that we've seen now in a number of these sorts of models, particularly for the motor cortex, but I don't think it's that special to the motor cortex, that the sort of the bulk modes of activity across the population often, at least sort of within a local operating regime, and here the two operating regimes of the delay period and the movement period itself, are often broadly linear. So we, for example, have fit models to these data which used a switching linear dynamical system model, where you're allowed to change the linear dynamics locally. And the idea there is that if indeed it's nonlinear, you should find different local linearizations automatically. And indeed, usually what happens in these models is you do get some switching, but it's almost all related to external inputs. And so in this model, we capture those external inputs by fitting this time series U of T. But we didn't allow that to be too complex. So the dynamical process couldn't come from U. U was just allowed to switch at the time of the go queue from one value to another value, but then that's the same value.
Yes. Okay, fair enough. So this is interesting to capture everything in the U variable. One question just disappeared, but while waiting for another question, is in the introduction you mentioned, like those two classes of model, like phenomenological and mechanistic / biophysical model. What about normative models?
Yes,
it's not belong to a class of model.
Well, I mean, I think normative models are really when we talk about normative models, we're often talking about normative models of behavior of the computation itself. That is, what computation should be done in the circuit in order to achieve some goal. There is sometimes, I thought about putting on that slide what I might call a theoretical model, which is sort of if I were to build a circuit that did this, how would I build it? Kind of model. In a sense, I mean, that's a hypothesis. Perhaps, the word model, I think, gets a little slippery until we bring to bear a data-driven approach or a more mechanistic approach. We don't have any real reason to think that that sort of hypothesis is a description of the real system, right? So I was thinking more about models that are really trying to describe the system in some way, rather than models that are thinking about how the computation could be implemented in principle. Obviously, we do all of these things and try to connect them in some way.
Okay, and maybe just a last question from André Lantin. So what are the dimensionality constraints for the network size, so the W?
So the dimensionality of the actual EI system, I mean, we've explored things up to about 700, and that's largely just sort of keeping the covariance of the memory of everything in memory successfully kind of constrained. I'm sure we can scale that up further, but it didn't seem really to be necessary to push that up very far. dimensionality of the recorded data, so there's about 700 recorded neurons, but the systematic dynamics, so the variance captured by principal components, is concentrated in about 20 or so. And so that's about the order of the random projection that we choose in J.
Okay, great. Thanks a lot. Thanks again, Maneesh. That was an absolutely brilliant talk.